We now turn our attention to producing food in precision fermentation with our next guest, who is a biotech expert with 19 years in downstream processing and project management. He has been leading plant biotech at the Fraunhofer IME since 2019. So please do join me in welcoming our next guest, Dr. Stefan Rasche, the head of the Department for Plant Biotechnology at the Fraunhofer Institute, Germany.
Thank you very much for the kind introduction and also for the invitation. So we have already heard a bit about the term precision fermentation or precise fermentation, fermentation in general. And before I come to some precise products, I would like to introduce you to the topic itself and define what is precision fermentation and how does that differ from other fermentation processes. So it was surprisingly difficult to find out what is precision fermentation in detail, because it's not really clear when this term was emerged for the first time and what it precisely means. But if you look at Google and alternative sources, you find a lot of different definitions, and they all have a certain theme in common. It's about precision in certain things. So one says it's about the design of a particular end product. It's the design of the process. It's the use of intelligent methods to boost the process or the product itself. And a very good combination of all those things is a statement from Professor Wittmann from the Saarland University, who says that it integrates custom cell factories, so the microbes, together with tailored bioprocesses, so including both the molecular but also the technical elements of the whole process chain. And this is also what the scheme shows us. So we are starting with the strain generation and improvement, going on for screening, move on to process development in terms of fermentation, and then process development in terms of downstream processing, and later on combining that all into a scale-up. So when we are designing a precision fermentation process in our labs, we usually start with a more or less general theme. We start using different genetic engineering and also digital models to improve the target protein structure on a genetic level, or also for the host, wherever that's applicable, and then move on to target-specific assays to screen if our improvement has really led to also an improved process, which can be activity, which can be yield, growth rate, whatever. So then we move on to small-scale parallel fermentation in combination with experimental design to fine-tune the process of growing the microbe to really get the highest yield out of it. And then the next step would be the downstream process development, where we combine harvesting, extraction, clarification, and purification methods all together to find the sweet spot on all these different techniques to get out the best yield. But what comes on top, so that's all the, let's say, technical and biotechnological things, is the regulatory level, which is also extremely important and often forgotten or addressed too late. So the first question we have to ask is which production host can be used to later produce our product? So that's quite easily to answer. If we are in food, then it's potentially an organism on the GRAS list or on the QPS list. But what else is to consider? So that can be any purification tags that you add to your protein that works nicely in the lab. So everybody in the lab is using his tag. This is great. This can also be potentially scaled, but then causing issues. Next thing, and this is a very interesting and important issue when dealing with proteins that are associated for the food later on, do we need sensory evaluation? So has anybody to taste the protein as part of the sensory evaluation? Because to do so, you have to provide the safety of that product at a certain stage with a certain level of details, but you also have to produce enough to either do the safety studies and the tastings. And this is having a great effect on screening. And next, there's chemical compounds that have to be addressed, media, all kind of techniques that have to be addressed and made to be sure that these are, in principle, also scalable. So there's a lot of additional challenges that comes with precision fermentation that I'm not going to talk about, because that would take very long. But one of the biggest challenges when you look at different companies is the regulatory framework. So what is necessary to provide to the authorities to get my product approved later on? And with all that in mind, we have to ask ourselves the questions, how do we get started if we want to launch a new product? And unfortunately, there is no one-fits-all solution. That would be too easy. But there are a few general things that can be considered when getting started. So first of all is, what is my product from a regulatory perspective? Sounds also easy, but especially in terms of wheat proteins, it's not so easy. So it could be novel food, it could be sweetener, it could be artificial, whatever. So it's not so easy to answer that question directly. So next thing, is my plant process food grade compatible at all? So this is also something to make sure as early as possible before you enter in certain surprises, like you will also learn later on from one of our examples. And what kind of safety assessment is required during early development phase. So as I said, there is no one-fit-all solution, and of course no one example that I could show you. I picked up 3 projects from our lab that is touching precision fermentation in food. And to tell you a bit more about this, where we failed, what we have learned of it, and where we are now improving. So the first project is called Phe-free. It's about the dietary management of phenylketonuria using a novel diet containing recombinant Phe-free proteins. So that was our first project in that field. And just to remind you, phenylketonuria is a genetic disorder where people have a genetic effect on the phenylalanine metabolism. So one enzyme is malfunction, and they cannot rightly process phenylalanine. And that can lead to severe mental effects as the byproducts are accumulating in the brain, and this is really having severe effects. So, and the most, let's say, prominent cure for that is a lifelong strict protein-restricted diet. So in some cases there's enzyme replacement therapy, but in general it's a lifelong diet. And so that means that you are not allowed to eat food that is protein-rich, and all the essential amino acids that you are missing are supplemented with artificial amino acid mixture, and they taste really, really, really bad. So the aim of this project was to produce as close to nature protein as possible that has no phenylalanine, but all essential amino acids in a good composition, and that can be produced recombinant, and that's what we did. So we selected from more than 800,000 protein sequences, just a handful of sequences finally that met our criteria, and the one of them, GSP 105, was finally produced recombinant in Pseudomonas fluorescens with very high titers, can be easily purified with good purity and yield, and we also have a preliminary classification as a novel food, which is also pretty important. So all animal experiments done so far are proving that the protein is efficient in treating or in managing PKU, and right now we are a bit stuck in the phase of scale-up, and why so you will learn later. So the first thing that we learned from the process is that the regulatory approval is a very crucial thing that you should address as early as possible. So it took us more than 3 years when we had the final protein to get the preliminary classification as a novel food, which is in the end not much worth. So if we look at the case of Impossible Food, they have their products been fine on the market in the US, but regulatory approval in EU was still pending. So, and I'm a bit afraid that we are running in the same direction with our protein. So the second thing is more a technical issue. In the beginning, we had, like always, used IMAC purification. So that's a classical purification technique in molecular biotechnology. And it worked fine. And we were also sure that we can scale it. We had all the resins bought with appropriate quality and so on. But later on it turned out, well, yeah, you can in principle scale it, but there's no company dealing with the large amount of heavy metal ions that you will need to charge your resin. So even if it works, nobody will take it because it's too complicated. You have to deal with too many restrictions and so on. So in the middle of the process scale-up, we had to change our entire purification strategy, which luckily worked. So we moved to ion exchange. Everything was fine, we thought, and then when we entered the real first interaction with our final CMO company, they told us, well, the buffers you are using are not food grade, so we can't buy these substances in food grade quality, some things that we did not consider a year before, but we will consider that in all the future projects for sure. So that's something to learn from that. Check all your materials, if they are scalable, if the companies are also willing to scale your processes if you take special materials, and check that all your buffer components are really available later on also in multi-ton quantities. So in scale-up was also an extremely different topic for itself. So first of all, it was hardly impossible to find a company that was interested in running our project. First of all, there are not many CMOs in the, let's say, volume range that we are looking on in Europe. Just 8-9 different companies that have the fermenters in scale and have food-grade permission and so on. So that was already some kind of bottleneck. And on top, our process was just too small for the larger players. So in the end, we were addressing a volume range like 500 to 1,000 cubic meters a year, and that's not much for the big players. That's too much for the small players, so that was a problem itself. And finally, when we had our CMO, in the end, we could not transfer the process really well because there was a clear bad communication when we are talking about what is necessary to provide from lab to the CMO to get a process scaled. So here's also the clear recommendation: get in touch with your CMO as early as possible and check all the details at least twice before you run into surprises. Okay, the second project is called Novel Sweets. It's about improving sweet proteins. So we already have heard a lot of background information, what sweet proteins can do and what they can be used for. So I'm not going to talk about that. The project was aiming about not to produce the natural proteins, but to have an improved versions. Because all these natural sweet proteins have one drawback: they usually don't taste as well as you would like to have them. So taumatin, for instance, has a liquorized aftertaste, so it can only be used for some very special applications, and the same is also true for most of the other proteins. So the aim of the project was to improve the sequences with different molecular docking methods, to work on the aftertaste, lingering aftertaste, sweet profile, off flavor, and so on, and simultaneously increasing the sweetness just to reduce cost in the later production, because the sweeter the protein, the less you have to use later on. So the project started in 2021 after a quite long application phase. We had several rounds of optimization cycles when we were addressing the sweetness and the taste profile of the protein, and finally end up with a version called X3. So that's roughly 10,000 times sweeter than sugar on a weight base. We have to improve a bit on the lingering aftertaste still, but the candidate is not so bad at all. And right now we're in the phase where we are formulating that into model products and looking for companies for scale-up production. So what we have learned here is the regulatory approval route is extremely important. So there's a lot of different options that we have, and to make that short, because time is running, we will probably go via FDA first, because in Europe it's hardly impossible to get anything approved via EFSA in a short period of time. So that is also something to keep in mind as early as possible. Where do you want to apply for your first approval, and then take all the information that you got from that process and move it to the other countries. So the next thing, and that was extremely important in our project, is the taste and safety assessment. So the most important criteria for our optimization next to the production level was how good does the protein taste. So, and there is no artificial method to taste or to sense that. You have to taste it with a human panel. And then comes the question, okay, what kind of safety do I have to provide for tastings, not for eating it, so just for tasting? And that was also difficult. So of course we put it into our application, but finally it was then, after it was granted, rejected. So we have to work on it and improve it again and again. And The proteins are extremely sweet, so you don't need much. You can produce it in small volumes during the optimization phase, which is awesome, but you need a lot of material to do the safety assessments, not necessarily because you need it for the test as such, but the regulatory framework says, well, you need to have to provide at least 1 gram batch from which I take a couple of milligrams and go on. And this is causing a severe issue if you have a limited budget in your screening phase. So keep that in mind. So as an outlook, this is a completely, let's say, off-topic thing for the novel food things, but we are also working with plant cell cultures in our laboratories and have developed a system where we can modulate the metabolic profile of plant cells just by using different light recipes. So this is a very interesting technique, especially in Europe, because we are not using GM techniques to boost a natural plant cell system, but just light, and thereby avoiding all that issues associated with GM, mainly on the customer acceptance side. So this is also something just to keep in mind, that there are more ways to improve a precision fermentation process just by genetic engineering. There are also environmental factors that you can consider. So, and that's the last slide. So to summarize all these things that we have heard before, so as there is no one route to follow, but there are a few general things to consider. So first of all, what is the regulatory perspective of my product? So make sure that you know which route you want to follow. So do you need tastings? If the answer is yes, then consider this as a very, very important part that causes a lot of money and time. Do you already have an idea about your upstream and downstream process? So check that all your material is scalable and compatible with your approval route. What do you need for a successful scale-up should also be known. And the last thing, if the use of genetic engineering methods will be a concern for customers, like in cosmetics, where any kind of GM techniques are absolutely red flagged, think also about alternative ways to boost production, if that's possible at all. And with that, I would like to thank for your attention and